procedure for the transmission to the enucleate metaphase 2 oocyte of the DNA nucleus of a somatic cell for a new person's generation which is genetically identical with the somatic cell donor is somatic cell cloning and nuclear transfer. The achievement in the cloning of a whole mammal dolly from one adulthood epithelial differentiate cell triggered a scientific revolution. It has shown that tissue differentiation genes inactivated by nuclear reprogramming may be totally reactivated while the reversal of a differentiate nucleus to its diploid condition. Somatic cloned animals may be utilized for numerous copies of genetically elite agricultural animals for the creation or xenotransplantation of transgenic animals for pharmaceutical protein or for the preservation of endangered species. It also offers huge biological potential for therapeutic cloning and allotransplantation with optimization. Cloning has not only become a key technique for the research of gene function, genomic impression, genome reprogramming, development control, genetic illness, and genetic therapies, but many other subjects as well. Cloning is the most recent advancement in animal husbandry selective aided breeding. Cloning animals is a dependable method of generating superior livestock genetics and ensuring that herds are kept in the best possible condition. It is vital to realize that cloning doesn't affect an animal's DNA or affect its genetic makeup. It's just a different type of assisted reproduction. Cloning allows livestock farmers to make a genetically identical twin of an existing animal.
Cloning process. The first step is isolation of DNA gene. The plasmids and DNA gene are taken from its cells. Then, by using restriction enzyme, a specific restriction site will be cut and it will generate a DNA fragment either with a blunt end or a sticky end. The second step is the selection of suitable vector. A vector must be able to self-replicate inside host cells and possess restriction site for restriction enzyme. An example for a suitable vector is plasmid. The third step, the insertion of target DNA gene into the plasmid vector. The plasmid is cut open by the same restriction enzyme used for isolation of target DNA gene, and with the presence of DNA ligase, the base pairing of target gene and plasmid occurs, forming recombinant vector. The fourth step, transformation of recombinant vector into suitable host. The recombinant vector will be transformed into a suitable host, mostly a bacterial cell. This step is done in order to replicate the recombinant DNA molecule in order to get multiple copies of gene of interest. And the last step, isolation of recombinant cells. The recombinant cell is then grown in culture media, but the culture may contain both recombinant cell and non-recombinant cell.
Take this for example. A successful cloning was done by scientists to produce dolly, which is the first mammal to be cloned from an adult cell, not from embryo. The first step is, the nucleus of another cell was taken from a fin-dorsate white sheep. From the Scottish black-faced ewe, the nucleus is removed from its unfertilized egg. The other cell is then inserted into the cell that have no nucleus. When the insertion is successful, electrical pulse is used to make the cell and eggs fuse together. The resulting cell will be cultured outside for about 6 or 7 days for it to divide and become an embryo. The embryo then will be implanted into a surrogate mother, which is another Scottish black-faced ewe. A white sheep face, or base known as dolly, was born.
Cloning is the most recent evolution of selective assisted breeding in animal husbandry. Important to remember that cloning doesn't manipulate the animal's genetic makeup nor change an animal's DNA. First of all, the benefit of cloning is allow us to balance environmental habitat, where cloning can be used to protect endangered species. For example, in China, panda cells are being kept on reserve should these species numbers be threatened by extinction. Imagine that if an animal becomes extinct or disappears because their habitat does not support life in needed ways, then the outcomes in the local region can be very dramatic. Furthermore, Cloning can create more security in the global food supply. For your information, cloning animals is a reasonable approach that could help to stabilize our supply of animal-based protein. Most of the foods from cloning will be from the offspring of clones that are not cloned themselves but sexually reproduced animals.
The cloning of animals has been occurring for a number of years now. Although there are clear benefits to humankind, I believe there are also disadvantages of cloning technology. First and foremost, cloning process is expected to bring new disease. One of the real possibilities of cloning is cell mutation, which known to result in new and more aggressive genetic disease to begin. Cloned animal can experience life-threatening health issues at any time because the cell programming can alter their genetic profile. This process can reduce reproductive capability in some species, and some outcome doesn't have a known cause. Next, cloning can cause diversity loss, eventually changes the species anyway. The term genetic bottleneck is used to describe the loss of diversity within a specific species. A lack of genetic variation creates a reduced resiliency for the offspring in that species. Lethal genetic disorders are more likely to occur. It can occur naturally. To eliminate diversity loss, a similar species would need to be introduced to the genetic pool, which would change the future course of the animal anyway.
Somatic cell cloning or nuclear transfer is a technique to create a viable embryo from a body by taking an nucleated oocyte and implanting a donor nucleus from the desired somatic cell. This laboratory technique used to obtain the patient-specific pluripotent cell for therapies and diseases research. Somatic cell is used because this cell contains inner part information of the subject. There are three types of cloning. First is gene cloning that copy the segment of DNA strand. Second is reproductive cloning that used to clone the whole subject such as animal or human. The last cloning type is therapeutic cloning that clone the embryonic stem cell used to creating new tissues for injuries or diseases. Cloning process consists of 4 steps to declare the complete cloning. Begins with isolation process and followed by ligation that have 2 steps in it, that is selection vector and insertion of target DNA gene. The next is transformation process and the last is screening process.